final presentation of the annual meeting. Mitch, for your team members, and we are recording these meetings, and we will post online links to the presentations, all of them. And we do have, we are willing to send them out by flash drive if anyone is not able to access and download the presentations remotely. So just let Christine know if you need any flash drives to anyone.
Sure, I forwarded the links to the earlier presentation, I think late last week, Thursday, I think, when we talked. And I think they both, Elizabeth and Usman both indicated that they had seen and accessed it. So that might work just fine. Maybe it's just a bad day.
Well, I think the global internet's been a little bit shaky. And I have to say, even last presentation with Jeff's, we were getting a little bit of unstable internet. So we'll do our best right now. So I'm going to turn the microphone over to you. And Christine will go ahead and mute everyone else. There's a couple of features. You can click on the mouse over the screen to the bottom. There's a participants box. You can click on that. It gives you an option at the bottom of that to raise your hand if you have a question. Use the chat box to submit questions as we go along. With that, go ahead, Mitch.
So that worked out nicely last time, and I didn't have it figured out until we were almost done. I've got the participants on a sidebar. Where's the show presenter view and show?
If you want to clear that, if you just mouse down to the bottom of your screen, you should have something pop up if you're looking for that chat box. If not, I can go ahead and read those to you.
If you see something jump up, don't hesitate. Can you see my screen now, everyone?
We do.
Okay.
Yes.
Please go ahead.
Okay. So I think we have Elizabeth and Cyril on the call from Senegal, and perhaps Usman will jump in later. If Usman jumps in, let me know, because I'll have to give his part of the presentation. I was thinking that he was going to give that. But if he jumps in, let me know so I can make sure that I open an opportunity for him to talk about the program in Niger. So before, as I was sitting here thinking a little bit about the project in West Africa this last year, I was I was thinking about the visit that we had there in October, and so I opened my book to since the Senegalese are on here today. So Cyril and Elizabeth, Nanga Def. How's that? How's my Senegalese?
Magnifique.
Ah, magnifique. Thank you very much.
Yes.
So we actually had a really nice visit in Senegal in late October, early November, and I have a page in my handwritten travel book that has my some of my favorite expressions that they insisted that I learn to make sure that I could communicate at least enough to be polite. So what I have here for today's presentation, I'm going to let Elizabeth present in the section about Senegal, and that's early in the talk. There's about 30 slides here. They're essentially lined up with a little bit of introduction, very little introduction. Most of it is about the two programs. The PowerPoints are essentially laid out based on the operational plan, and so if you open the operational plan, I'm going to be going point by point through the plan, hitting those items that are relevant to the first part of the project. And so this project is called Sorghum Trait Deployment Pipeline for Food and Feed Value. And that's compared with the project, the first mill project, it was called the Sorghum Trait Development Pipeline. And I forgot, Cliff Weil is also on. I'm not sure if you're calling in from West Lafayette or from Washington, D.C., Cliff, but he's the other Purdue PI.
I'm in Indiana today.
You're in Indiana.
And Mitch Usman is online now.
Okay. Usman, are you on?
Yes, Prof. How are you?
Excellent. Can you see my slides?
Yes, I can follow them.
So when I get to the part of the program about Niger, I'm going to ask you to present. And as long as the connection is good and we're able to, I'll let you do the talking.
Yes, I'm ready.
Okay, thank you. So this is a collaboration between Purdue, INRAN, and Sarah Sisra, and I've already introduced the collaborators, and now Usman representing INRAN. So the project has two principal activities or objectives: to develop and commercialize sorghum cultivars with improved post-cooking protein digestibility. Probably should have thought a little bit more about this. I didn't put in all of the introduction slides about why this is important. I just organized this around our operational plan. So for the external evaluation board, just a reminder, one of the things that's unusual about sorghum compared to other cereals, when you cook sorghum, or when you make a flour out of sorghum and you cook sorghum in an aqueous environment, the protein loses about half of its digestibility. And sometimes even more than that, depending on other kinds of secondary metabolites that might be in the mix. So in phase one in SMIL 1, if we want to call it that, or phase one of the SMIL project, the Purdue project, this same general group invested a fair bit of time with Elizabeth and Usman as PhD students at WACI. And Elizabeth's project involved screening a population of mutants looking for mutations that would restore protein digestibility, essentially. And so during phase one, we actually identified 3 mutations that essentially almost completely restore protein digestibility. So there's very little loss of protein digestibility with cooking. And in phase 2, where the objectives, and you'll see momentarily, really revolve around how do we use those genetic resources or those genetic tools to enhance the nutritional value of sorghum with regards to protein digestibility, particularly as it involves making products like tuo and other products where wet cooking is involved. And so activity 1 focuses on developing and commercializing sorghum cultivars with improved post-cooking protein digestibility. The first sub-activity, so activity 1.1, is to develop and advance breeding populations for highly digestible protein using the HL locus from P721Q. So when Elizabeth started her work in Ghana, we hadn't identified these mutant alleles that will be a little further down, 1.2, 1.3, 1.4. And so the first project that she started on was, let's use this as an example of how one would use this kind of a genetic resource to create sorghum varieties that have improved protein digestibility. Elizabeth, you've got about the next 15 slides. I think you've seen this presentation a few times. This is the one that's loaded is the one that you sent to me about 20 or 30 minutes ago. Would you be willing to go up through the slide in your presentation where I think it's lessons learned, or maybe it's COVID-19 adaptation or something like that? And I guess I'll have to probably forward the slide. Just tell me when you want me to forward the slide.
Yeah, sure. Thank you. Can you hear me well?
Very well, thank you.
Yes. Okay. Thank you, Prof, for that introduction. And as you mentioned, I'm going to go through the activities that were performed in Senegal, and part of the SMIL phase 1 was to introgress the HI locus into the elite Israel lines that were released in the past couple of years. And this led to developing 4 main populations, and out of those 4, 3 was at stage BC3F4. And in this phase 2, we want to, since we want to test them in multi-location trials, the idea was to advance them at least to BC3F6. So one of the activities was to advance these, and it is ongoing at Bombay right now. And also in the greenhouse at Sarah's, we are multiplying seeds as well as in Bombay to get ready for the next field season, to have enough seeds to take to the farmer's field in next field season. You can go to the next, please. Next slide.
I'm on, I don't know if you can see what I'm looking at, 1.1.2 test
okay.
value.
I think it takes some time before I see the slide, so it's fine. I will just.
This is the slide that has you. This is the one we're in Daru.
Yeah, sure. Okay, yes. So as I said, during phase 1, we were able to initiate those crosses, and one of the population that was most advanced was at BC3F6. So as part of my thesis, I was able to test those lines in the lab for protein digestibility and identify 18 lines that have high digestibility and high post-cooking protein digestibility. And they were from a cross between one of the 8 lines named Fauru, ISRA 621B, and P721Q. And these, during the last field season, they were tested in Bombay, Daru, and Sinchumalem, along with the 2 parents, P721Q and Fauru. Unfortunately, we lost the trial in Sinchumalem due to very low germination that occurred. So the results in Daru show actually 5 promising lines that combine high yield. That's in the next slide, please.
Yeah, I just forwarded. I'm on the next slide. This is 1.1.2, and it has.
It takes some time before I actually see them. So
I'm describing it to you because I know that you've looked at these. This is the one that has the matrix plot.
Yeah, sure. So what I do is that I have actually the slides on the side here so that I don't have to wait. So you can follow and pass here. So at Daru, where we tested this is actually trial, on-station trial. Let me precise that, it's not yet on farm. So on station, we're able to identify 5 lines that combined high yielding and high yield and also good resistance to grain mold, which being a must-have trait based on the breeding product profile that we developed for the sorghum breeding program at ISRA. And in Bombay, also same, the lines also showed some also good scoring in terms of grain mold. About more than 90% of the lines had good grain mold resistance. But when it comes to grain yield here in Bombay, we actually faced a disease infestation that started during grain failing. That's why actually we have here lower yield. Still, we found in the midst of that grain disease infestation, we have 6 lines that showed better performance in terms of yield compared to the rest of the 80, compared to the other 13 lines that were tested. And as can be seen in the next slide, on the left, we see how the field was impacted during grain failing. It was about 1 or 2 plants that were maybe infected in every other block. But by the time we reached maturity, almost the entire field was infected. And right now, we don't have much information about the pathogen that is responsible for this infestation. We're still waiting on the results from the phytopathology lab. And the lady that, the Mampenda, who is in the project with Clint McGill. And they are the one doing this analysis to get us feedback to understand what was going on. But even with this invitation, we had some lines that actually showed some promising yield performance. When we look at the combined result for both sides in Bombay and Daru, we actually see four lines that perform well both in Daru and Bombay, and those are for now what we select as promising lines, even if we still look at the other ones, but we pay more like close attention to these for the next field season when they will be advanced in on station as well as on farm trials. So after harvesting the seeds, yes, we look at, after looking at the grain, the yield potential as well as the grain mold and other traits, it was important to look at the digestibility in the lab for these traits. And we perform, in the third activity, we perform at in the lab in Ceras, which is a lab that was established during with the fundings from SMIL-1. And one of the main challenges that we faced with this performing this activity was that our hybridization oven broke down, and we were not able to really make much progress for this specific activity. But as we try to use some, come up with some solutions, which is using, for now, temporarily a water bath, which can give us the desired temperature at which we can cook these samples. We don't know yet if that can affect the results or not. That's why we are also working with the team at Purdue, with Dr. Weil and Dr. Tuinstra, to see how we can actually temporarily acquire some data from there, do a test there and acquire the data while we're waiting to fix the oven or actually get a new oven, since that's budgeted in the project. For the second activity under this objective was to introgress the highly digestible protein threads from the EMS mutants that were developed at Purdue. As Dr. Tuinstra mentioned earlier, there were 3 new mutants that were identified, and we were trying to, what we tried to do is to introgress those traits into the edit line at ISRA. And during my thesis, I was able to work with these lines and the resequencer resequence them, and in order to identify candidate SNPs, candidate mutations that were identified, and also those candidate mutants, how we can actually make use of that result by developing CASP markers that can help easily identify the lines that have integrated the digestibility trait and use them to really advance the breeding program more quickly. And that CASPR genotyping is ongoing. So far we have some result that we think is promising, but we also need to perform more and confirm what we have so far. And there is one student, the lady on the left, who is a master student working on that part of the project. I will introduce them later.
Can you say a little bit more, Elizabeth, about the markers that you used in the breeding program, essentially validating them in a breeding context? I know they haven't been highly validated yet, but what did you find so far with your use in the breeding program?
So these markers were developed from using the regions that surround the candidate SNPs that were identified after characterizing 2 of the mutants during the first phase of SMIL as part of my thesis. And so far the idea was to validate these in the breeding program. So we were able to validate them in the populations where they were, as I say, identified from. But right now, we haven't been very successful, to be honest. But as I said, there are some results that show, as I said, like promising results that we can focus on. And what we're doing right now in the lab, since it's really based on PCR, is optimizing the protocol to see how we can actually get better results. That's where we are at right now, but it's ongoing. It's ongoing.
Thank you.
Mm-hmm. So at Purdue, what was done was these mutants were crossed with elite lines, elite West African lines, and as part of Dr. Tuinstra's breeding program, and we received them, I think, last semester in Senegal in order to initiate back crosses and to develop, improve the population that have these alleles. And they were planted in September last year, in September. However, we, since it's off season, we mostly relied on the irrigation to perform this activity. However, the irrigation system was not the best during flowering time, so we actually faced a lack of pollen. And as we know, as breeders, pollen is crucial to be able to make crosses. So as a solution, we decided to make those breeding nurseries again during this off season, and since the irrigation system is working better this time, they have been planted early March, and we hope to initiate those back crosses in the next couple of weeks. So as part of this project also, we work on training and capacity building. As I mentioned, I was trained during phase 1 for my thesis, but in this project we are actually, I, myself, and also Cyril, we are closely working on training 2 master students. And Aminata Saou, who is the lady working in the lab to perform the digestibility analysis, as well as validating the markers, the CASP markers in the breeding programs. And Jean-Noël Chow was the one that did the agromorphological evaluation of the lines in the multi-locational trial. They are right now based at CERAS. So far, the achievements that we have is that we have 4 really promising lines that have that show good adaptation in the field and also have good great food grade quality, in addition to being highly digestible after wet cooking. And these will be tested better in the second year. This field season, actually, hopefully this will happen, and to see actually how they perform well, to have better data to look at. And as part of the project also, we reinitiated, I would say, collaboration with ITA, which is the food industry in Senegal, because one of the objectives I think from year 3 is to see how these lines can be used in making bread, couscous for human consumption, and also work with industries like Sedima and Enema, who work on making on poultry feeding as well as also human consumption, to see how these can be used in making poultry feed. And also Mamel Jabot is also, these are some of the lead industries in Senegal in their area, which is animal feed and also human consumption. Mamel Jabot makes a good, what they call in Niger dege, we call here chakri. So we met them also with Cyril and I recently to initiate those collaborations to see, so by the time we come in the next couple of years with a product that is ready, they will be more open and willing to actually receive, because right now when we met them, they were actually already open to testing that. So this was a good, I think this is a promising collaboration, I would say. And when Dr. Tuinstra came here the past, I think fall, we were able to visit some farmers' field in the Thiès region around Mour. And where we met those farmers, they use the lines, the varieties that we are actually improving in this project. So they already accept them the way they are, even if they have low grain quality. Maybe they don't know much about the protein digestibility yet, but we're able to talk to them about that. And they already like these lines, and they grow them and they have good yield. And also what they told us is that the fodder, after they harvest the seeds, they take the stems and feed them to the animals, and they also sell it as almost the same price as the grains. So for them, it was really an important thing that made us really think in how to orient this project and actually use this line maybe in a dual purpose sorghum, maybe not in this part of this project, but looking for funds to integrate that, since it's very important for the farmers. They mentioned it themselves. So for 2020, what we plan to do, since we already met with the industries, Sedima was willing to based on the figures that we show them for the lines, their protein content and other nutrients they have, phosphor, since that's also important, they said for them, for the product that they make, they were willing to take these lines, but at first they want to test them first and confirm the figures that we already give them, which is good for us. So we plan to send them those seeds, and they will make the lab test for now. Can't use it yet for poultry feed until we actually develop better, more stable varieties out of this. And also the other activity we plan to do in this year is on station and on farm field trials to test these varieties in the farmer's field and see how they perform in reality, and perform the participatory varietal selection with the farmers themselves to select the best lines that we can release in the next couple of years, hopefully. So yes, we are right now facing the COVID-19, and we plan to use conference calls just like this one to plan the activities, since we can't do it in person for now, and the field season is really approaching very fast. And also, in order to do some of the activities like the field and the participatory the participatory varietal selection, we mostly rely on institutional guidelines to be able to perform those activities. So I think that's all for Senegal.
Elizabeth, do you have any sense of, I mean, essentially the pandemic is just getting started. Do you have a sense of how Senegal might respond to this? I mean, there's lots of different ways that governments are responding. I don't know how severe the case might become in Senegal,
Yeah.
but it's likely to have a major impact on research at Purdue. But I haven't had, I mean, that's essentially all I've been doing today is managing this issue at Purdue with our various research projects. I was just wondering what your thoughts were about how far things are likely to go in Senegal.
In Senegal right now, we are facing an emergency situation where we have, how can I say, a temporary lockdown just at night for now. But then during the day, we personally at ISRA, we are not, most of us are not going to work right now. We're working from home. So as far as how the government will respond, I really can't tell for sure, but we have 108 cases. Might seem little compared to other countries, but for us, it's really a lot, as we can see how this can spread easily based on our cultural things that we do here. So it's really more prone to spreading easily. So for now, as I said, we just wait on the guidelines, since we depend on the Ministry of Agriculture to do some of these things that we do.
Is it possible that they're going to restrict your ability to plant trials?
Maybe, maybe not. I'm not sure about that, since it requires people working together in the field. Maybe reduce the number of people we can work with, maybe that, but some activities maybe not. Like, as I said, participatory, like where we have to do some surveys and all that, might not be able to do that. We might not be able to do that, but we just hope that it will get better before that. But
in some places in the US, we actually are restricted to how many people you can have in a vehicle at one time. And so some of the offsite locations, it took us all day to get to some of these other locations. And it would be hard for me to imagine if you were to go with a team of 4 or 5 that you would take 4 or 5 different vehicles.
Yeah, here, you have to have maximum 3 people in a 5 passenger, like 5 people's car. You are only including the driver. So only 5 people, only 3 people including the driver. So yeah.
So before we jump over to Usman's part of the talk, does anybody have a question or questions or thoughts while Elizabeth's presentation is fresh on your mind, and then we can jump over to Usman's, which is the next activity.
Bettina's got a couple of questions. Bettina?
Yes. So thank you really for letting Elizabeth also to present. One question I have is, what is the actual target agroecology for those sorghum genotypes? Or could you explain the rationale for the choice of the test locations? I believe that Sinthiou Malem is really in a different agroecological zone than Bombay, for example. So what was the reason to choose this diverse set of environments, and what is the real target region you are aiming to breed for?
Okay, so the idea.
Let me answer this question, Elizabeth.
Oh, okay. okay, Cyril, if you want, yeah.
Okay. Currently, we are more working on the codatum types, and we focus on the zone compressed between 300 millimeter and 700 millimeter. upper these zones, we used to focus on the genotypes. When we have over than 700 millimeter, we precognize the genotype. But for this environment, we work more on the codatum types. So the limit is north of Sinchumalem. Just after Sinchumalem, well, yes, after Sinchumalem, the codatum type did not work because of grain mold and other things.
Okay, so it was just an extreme of your target in environment.
Yeah, but also these, okay. I just wanted to add that also the locations that were chosen are also diverse, as you mentioned. They also to be able to see how these lines perform in different type of environment, not just one, which is Sinchumalem, which is the wetter part, compared to Bombay or Daru or other areas in Senegal as well. The diversity was very important as well.
Okay, yes, I understand this. Given the high interannual rainfall variability, it's good to look also at the extremes. Now, my second question was on the variety E45 at Daru. It had a very high yield compared to all other varieties, like 4.5 tons. How do you explain this performance, and is it not an outlier? Did you estimate repeatabilities for the plot level yields at the two sites? Yeah, here it was like.
This is the combined analysis. Is it the?
Yes, but it still shows the high yield of.
This is Daru.
Yes, this is Daru, but especially also in the combined analysis, you see that it's really outside everybody else.
Mm-hmm. Yeah, I was even talking about this with my colleague earlier. Yes, these lines actually, it's not, how can I say, it's not really much surprising, because the parent being Fauru and some of the three other Isra lines on station, they can show really up to 4 tons per hectare. So this was a trial that was done on station. So seeing maybe 1 or 2 that perform better than others, having up to like 3-4 tons per hectare, I think is possible. But as you said, also these are results that we are also still exploiting as well. And I will pay close attention to this specific line, E45.
Yeah, no termite hill on this plot or something
Which
like
plot?
that. Just to be sure, because like at Bombay, it wasn't performing so well.
Mm-hmm.
So.
Yeah, as I explained, Bombay, because we had the disease infestation that really affected all the lines' performance. None of the lines really had a higher yield than 2.5. That was the highest yield that we saw at Bombay. But really in good conditions, when these lines are given good condition, at least the parents were seen to give a high yield on station so far. And as I said, we will test them on farm to see what is their true yield potential when they are in the farmer's field.
Okay, yeah, just be sure that your data is not biased by some outliers. Yeah,
sure.
but thank you. I get your explanations.
And on that previous slide that you showed, Danielle indicates that E113 seems to be a nice source of resistance for whatever that disease is in Bombay. There you go, right there on that far right-hand side. It's something to keep in the back of your mind once you determine what that disease is.
Yeah, that's very troubling, Tim. Actually, some of the early guesses by the pathology group was that I think it was macrophamina, wasn't it, Elizabeth and Cyril?
Yes. Yes, we were guessing, yeah, I'm guessing that it's macrophamina.
Okay. But we pulled up plants and we dissected them and we looked at the roots, and I didn't see the symptoms of classical macrophamina that I would normally. I'm not a pathologist by any stretch of the imagination, but I have worked with charcoal rot quite a bit, and I didn't see any of those classical signals. And the fact that the whole field was being affected, starting out near the road or the front of the field, progressing to the back, was troubling. Just trying to understand what that disease was.
Right, yep. Good,
Okay.
additional questions on the
Also,
clinical?
what I can add about this disease also, when Prom was here, we visited this field and he confirmed that he don't know, it is not macrophamina. So we are really, we don't know. I asked also Njaga Sise, he don't tell us which kind of disease we are facing. So we still using the phytopathology lab to see if we can get fungi that are responsible for this disease. This is our biggest equation for this disease. We don't currently characterize this disease, but I hope it is not macrophamina.
Great, additional questions on the Senegal work? Well, if not, how about we move forward then, Mitch?
We can't hear anyone.
I don't know, are you guys, is my participant window up? Can you see that, or is it just my screen that you can see?
Each individual will have to open up their participant screen.
In mine, I'm leaving it open so I can see who's on, but I can close it if it's blocking the view from my share screen.
All we see are your slides, Mitch.
Yes,
Okay.
all we see is your slides.
Okay, all right. Okay, activity 2 is around the Niger side of the project. Usman, are you still connected?
I think we may have lost him.
Yes, we did.
I don't see him on anymore. Yeah.
Yeah, he goes on and off. I think he keeps trying, but that's probably not easy.
What would you like to do, Tim?
Someone is trying to connect back, it looks like. Nope, that's Njito.
Yeah, now he's back on. He's what's at the bottom of the screen. User is what he's denoted as.
Oh, I see, I see. Usman, can you hear me?
He was on as Usman earlier, so I don't know if that's, if user is somebody else, Tim.
I asked him, he came on as user, so.
Okay.
He might be jumping in and off either on his phone and trying other ways to connect. We can wait a few minutes. I'd like for him to be able to present his own work, or I can present. That's either way is fine.
You can give him 30 seconds or so. Minute. I'd like him to present as well. Seems a bit stuck. Oh, are you there now, Usman? Mitch, why don't you start with the introduction?
Sure.
We'll see if he can.
If anybody sees Usman jump on while I'm talking, please feel free to interrupt me. I want to let him talk as much as he can while he can.
Yeah.
Okay. Activity 2 is to establish a forage sorghum breeding program at INRAN, and particularly at the Kollo Research Station. So when we had our project meetings last spring, I believe it was, April maybe, we were at the Grand Hotel in Niamey, and the Kollo Research Station, if you follow the river, the road that hugs the river, sort of to the south and the east, the Kollo Research Station is about a 25-30-minute drive. It's not that far, but the road in town is relatively congested and not in the best shape. It's a slow drive. It's not too far outside of town, and so this is a station that historically, in terms of the sorghum program in Niger, has been very important. This is a station that Issafu Capron started at. Its value proposition is that it is close to town. So when he's pollinating, he's close enough to town that he can either take a car or take his motorbike back and forth to work. He doesn't have to be separated from his family if he wants to be active in the breeding program. And Usman absolutely wants to be active in the program. He doesn't want to run his program necessarily through technical support. And I really respect that. And the fact that he's able to personally work in the field and direct the research program while he's able to spend time at home is, I think, I was really pleased to see how this is working out. The Kollo Research Station, let me see here. The Kollo Research Station had kind of, I would say, maybe fallen into disrepair, maybe not as, there wasn't as much excitement, or there weren't a lot of active research activities going on at the Kollo Research Station. But Usman had been in contact with Issafu Capron, and Issafu had encouraged him and described the merits of that particular location. And so he had really taken the lead in trying to re-energize and rejuvenate the program and working with the station director. And so in the last 12, 15 months now, he's actually did a really nice job of establishing a new sorghum program in Kollo. He has a nice laboratory. I'm not sure, about 12 months ago, when you would visit the Kollo station, they would bring you to a lab that I was not particularly excited about. It required a lot of repair, and it wasn't clear to me that there was enough enthusiasm in the program to really repair, or to get that sorghum lab in good shape. But about 10 months ago or so, something like that, last summer, they identified a different lab in the same complex. It's much better shape. The power works, and it includes a dry lab for seed preparation. There are 2 offices, one for the breeder and one for technical support staff, and then a 4th room that is being reserved to serve as a future cold storage facility. And so, again, in this case, having a sorghum research program within driving distance of Niamey, that's where the breeder, the scientist, the PhD level person is actually able to spend an appreciable amount of time in the program and directing the program, to me, is a very important situation. The program has all the basic equipment for managing the breeding program. When we were there, we spent a fair bit of time talking about his equipment needs. They're not substantial. The single most important thing that I've been asking him to work on is in terms of the breeding program data management system, and he's committed to the BMS system in terms of trying to use BMS as the default system for his program. So the station, the research station, the offices and the lab are up on the hill up above the Niger River, and the experiment fields are down below, adjacent to the river, where they can flood, irrigate. And so they have a breeding nursery within sight of his lab. It's about a mile, maybe a couple of kilometers away from the lab, within an easy drive. So again, someone can stay and work in the lab in the mornings and in the afternoons, or early mornings and later in the afternoons. And when it's time to be pollinating, it's easy access down to the field. In the last time I was there, the site is very fertile. It's all of the river bottom there is being used by farmers to grow different kinds of crops. So it's a good fertile field, but one of the challenges is that it's prone to flooding. And so when I was there in October last, he had actually had to build a berm around his field to keep the water out, not a berm around the field to irrigate and keep the water in. And so some modification might be needed in terms of moving the field. It looked to me like it needed to be moved a little bit to the west to get out of a flooded area, but maybe that's the last time it'll ever flood in the next 100 years. I'm not sure, but it turned out that last fall it was a challenging situation keeping the field dry. So the first activity beyond establishing the forage research program is to develop open pollinated varieties. And so Usman has been, Usman, just like Elizabeth, just finished up his PhD at WACI. He spent about a couple of different times, maybe a sum total of a year thereabouts, at Purdue working on this project. The interest was, how is the U.S. forage seed industry technology, or what's the state of the U.S. forage seed industry, and how might that be introduced into a program like in Niger, into a locality like Niger. And so there were two components of that. One of those included using the brown midrib loci, which are, these are genes that are involved in lignin biosynthesis. In particular, BMR6 and BMR12 are the ones that are used commercially in the United States. His PhD dissertation was actually involved looking at a number of different brown midrib loci that haven't been tested as well as the BMR6 and 12 locus. He's working with those in research mode, but in terms of his breeding program, he's using the same loci as that are commercially important in the United States. And so he developed a series of backcross populations as part of his dissertation to look at what are the effects of these BMR loci inside of Nigerian adapted germplasm. And so there were two lines that he was working with, or open pollinated varieties that he was working with. One of those is El Mota. That's a it's tall, a little bit higher rainfall adaptation, large seeded, a typical Mota type, but a Nigerian landrace, and the other is CPON 82, and so this would be an improved Caudatum type. So the CPON 82 types are often, or CPON 82 is very commonly used in their value add food science program. So all of the food quality grains, a lot of that work is emphasized CPON 82. And El Mota would be common in many farmers' fields. And so those represent two different kinds of sorghums that are being grown in farmers' fields. And he had backcrossed the BMR6 and BMR12 loci from US donors. And so early on, the first year he was working on this in his PhD, I sent him a bunch of donor lines that he could use in his program, and he backcrossed those into CPON 82 and El Mota backgrounds. And he was essentially using pedigree-based breeding approaches to combine stover quality traits, which would be the BMR and stay green type adaptation, with performance traits based on high biomass yields. He was also, that was worked directly inside of his PhD dissertation, where he was comparing near isogenic lines for the differences in nutritional value and biomass productivity. These would be near isogenic lines for BMR6 and 12 inside of those backgrounds. He also did straight forward breeding using a larger array of either improved or locally adapted guineas and mostly Caudatum type. Some of them are Federitas backgrounds, as well as things like Wassa would be an example of a guinea, where the BMR6 and 12 traits were being crossed into these locally adapted types. And then again, using pedigree breeding to combine the BMR traits with the local adaptation and stay green phenotypes that come with working in those backgrounds. And the last component of his research focuses on dual purpose grain sorghum hybrids, Sudan grass hybrids, and forage hybrids. And so this is really about a hybrid forage breeding program, whether they're Sudans or straight forage types. And this is as a counterpoint to the OPV type focus of 2.2. And so one piece of this involves, Niger has actually been quite active, especially when Issoufou was running the program 25 years ago, in hybrid breeding. And there was a fair bit of work that had been done looking at different kinds of seed parents that would produce hybrids that were adapted in Niger. One of the better seed parents that they'd ever identified was a line called N223. And so N223 is a B line. And so we took N223 and we crossed it with TX631 BMR6 and with TX631 BMR12 and developed some large breeding populations. And in the end of phase one, we sent those to Soumana Soleil in Niger who advanced those by pedigree breeding, so that he had essentially these tropically adapted caudatum type B lines, N223 by TX631 B lines that were either BMR6 or BMR12. At the end of SMIL 1, we transferred those over to Usman, and so now Usman has this collection of B lines and partially converted A lines, and he's continuing to backcross those B lines and using AN223 as the source of A cytoplasm.
Hello.
Usman, we can hear you. Just barely, though.
Yeah, so can you hear me, Prof?
I can hear you. How about the rest of the people on the call?
Just barely,
Yes.
but yes.
Mhm.
Usman, can you see the slides, or are you just calling in?
Oh.
So sorry, I was out. My internet was very unstable. So now I can present.
Okay.
I can see that we are at the seed parent development section, right?
Yes. I'll drop off. You go ahead and pick it up from here.
As you know, can they hear from me?
You cut in and out, but
So this activity actually started during the SMIL phase 1. Hello?
Hello.
hello.
Yeah, so I said that this activity started during the SMIL phase 1. So Sumana was the firstly responsible of this activity. Now that he has retired, he gave me the seeds to continue the back coursing program. We are actually doing the back course to get the BC3 and have the seed parents BMR6 and BMR12 line. We have identified good pollinator. So this activity also, oh my God.
Uh-oh.
It sounds like he dropped off. Can someone see on the participant screen, did he drop off?
He did, and he's trying to get
Yes.
back on, I think.
Oh, okay.
I'm sorry, bear with us here.
Okay,
There he is.
can you hear me now?
Yes.
Yeah.
Hello?
Yes.
Hello?
So, okay, that's good. So I said that this activity started during the SMIL phase 1. So now we are actually at this exact moment, we are doing the back coursing to get the BC3 line.
Usman, how many B lines do you have in both groups, the B6, the BMR6, and the BMR12 group? How many different B lines do you have, or the AV pairs do you have representing each background or each mutation?
I think we may have lost him again.
Yeah, why don't we just go ahead and we can maybe just get some comments from him at the end if he can get back on.
Okay.
He's fading in and out. So let's just go ahead and finish up and maybe he can just tighten up at the end.
Sure. So when I first started this, I made the cross and I produced several hundred BMR F3s in each population. I think I sent 100 of each to Sumana based on what he said he could handle. I suggested that he select for BMR. There's a lot of variation in terms of background stability and performance with these BMR traits. And I asked him to carefully select for those families that produced vigorous and well-adapted genotypes that had seed parent characteristics, meaning the value is that they have to be used as seed parents. And so you have to they have to produce large seeds, good exertion, normally large panicle types. These are going to be a white tan by a white tan cross. All the progenies are white tan, either BMR6 or white tan BMR12. And the intent was to be able to use these, both of those parents combine well with many of these other genotypes that we're going to talk about in the next activity. And so the goal was that he would be able to have 80 pairs, his own Nigerian adapted pairs, that could be used as testers of males or pollinators coming out of the next activity. And when I say brown midrib, the reason I put that picture there, if you look at the midrib of that sorghum line, you'll see that it's tan or brown. If you flip the leaf over, you'll see that there's a brown stripe on the bottom of the leaf. And that is a diagnostic phenotype for this mutation in lignin biosynthesis. So if you're going to develop hybrids, you need to have three different kinds of lines. You need to have A and B parents, which would be what we were talking about on the prior slide. The A's and the B's are the C parents, and you need to have pollinator parents. And so again, one of the advantages of knowing a little bit about the hybrid development activities that Isafu was involved with was knowing the parents that combined well in terms of producing hybrids adapted in this environment. So we leaned back on that understanding. I went to school with Isafu in the 90s in my PhD program while he was getting his PhD, and so I happen to know those pollinators well. And so the focus was really about trying to produce BMR versions of these things that could be used of hybrids that we knew already had good adaptation, really is what it comes down to. So we made a series of crosses with known pollinators that had both caudatums and guineas that would fit into different agroecological zones. We sent them to Usman this was all different kinds of complex pedigrees, but some of them were straight pedigrees and some were backcross ones. And that most of them were F3, F4, or BC1, F3, F4 in different combinations. We were curious that in the developed world, so a lot of emphasis has been made on developing dual-purpose hybrids. But if you go to developed world agriculture, in many cases, it's hard to have a dual-purpose hybrid that really is truly dual-purpose. It's usually it's hard to get sometimes forages that have good grain yields, or to get good grain hybrids that stand and produce good forage quality. And so in the US, most of the work has been on developing dedicated forage hybrids, and especially in the case of sorghum sudan grass types. And so we also sent them a few sorghum sudan grass pollinators, and one of those pollinators appeared to have very good adaptation based on our observation trials last fall. And so we're developing additional back crosses within the caudatums and additional back crosses within the sorghum sudan grass types. And we're intending one of those is as soon as we get the seed ready in the greenhouse, that we'll be sending to him, which is a BMR6 tan plant sudan grass pollinator. And this would be a dedicated thin stem narrow leaf type that could be used in grazing and in haying situations, and not unlike they're being used with some of the dairy programs that are there, having dedicated forages for them. So out of that large collection of R lines, BMR R lines that I sent to him, he made selections for things that had good adaptation, and he crossed 121 of those to AN223. So he didn't have the finished, fully sterilized versions of the BMRs, and so he went back to the original N223 parent to get a sort of a proxy understanding of combining ability. And so now he has a large set of hybrids that he's going to begin looking at in the coming rainy season in Kollo and in Maradi, so South Central, and so they're both kind of South Central, I guess. And this is a picture on the bottom here at the Kollo station of him making test crosses between the BMR males and AN223. So in terms of his program, one thing that's not listed here. Usman,
I am just back.
no problem. I'm on the achievements and lessons learned slide. Can you talk about that?
So I can go. Okay, yes. So for the achievement and lesson learned, I can say that the OPV development is really in a good shape because we have a lot of promising line. We've selected a lot. At the beginning, we have few entries. As we've developed we did a lot of courses. Now we have a large population and we are advancing them at Kollo now. The only problem we've faced was the reduced staff. As you know, in INRAN, we don't have a lot of technicians. So, and Sumana has retired. So I have only one technician who is working with me, and Kollo is not in Niamey. Our field is at 30 kilometers, so we have to be there early in the morning, and we've realized that the staff is really too small, and this is a big challenge for us. So for the rainy season, we are planning to recruit a lot of students in to help us data collection and conducting all the activities. Did you hear me?
Yes, I did. Very good.
Okay, so we can move to the.
I'm on the next slide now. Future
slide.
planning for 2020.
Yes. Thank you. I got it. So for the future planning for the 2020, we are planning to conduct on-station nursery trial at Kollo, at Koni, and Maradi. In my initial plan, I also added LOSA because I conducted a survey during my PhD at in this location. But due to insecurity, you know, LOSA is in Tilaberi region, and due to insecurity problems, that's why I discarded LOSA to this program. So we'll do it only in 3 locations at Kollo, in Koni, and Maradi. So we are breeding for farmers and stover traders. Their feedback is very important for us. When I did the survey, they mentioned specific qualities such as high biomass and stay green. Those are important for the farmers and stover traders. That's why we are planning to unseat the lines that meet their preferences. That with Professor Tuinstra, and during his visit in Niger, we had the opportunity for a lot of plans that we will, a lot of activities that we will conduct during the rainy season. So the main locality here that we've added is Gaia, because our collaborators in Gaia are well aware about the activities I am conducting here, and they had feedback from, how do we call them, the local farmers, and they are actually interested to the new program we are running. So in addition to what we have achieved, as soon as the off-season activities are done, I will go to Gaia to have a survey discussion with farmers in that locality.
I just forwarded the slide and we're now on expected output.
You, yes, we think that after achieving all those activities, the best BMR PVs and the hybrids that have the potential for increasing food and feed security and household income will be selected during the on-farm evaluation. And at INRAN, we have the laboratory for nutrition analysis. Lana, the one you visited in INRAN Gamkale last time. And we are planning to do the feeding analysis, the nutrient composition analysis at the Lana for all selected lines. I'm not sure you got me.
Yes, no, we heard you.
Okay.
I just forwarded the slide to contingencies and COVID-19 adaptation.
So, as you are all aware, finally the coronavirus was reported in Niger, and we are following with great attention the news about this disease, and our main guideline here is the government's orientation about this disease. At the office and even in the field, we are working with technicians and farmers who are helping us currently on the field. So we are taking the the strategy adoption and all the management that must be done are being done right now. Everybody is protected before starting the work in the office and on the field.
How's that being done, Usman?
So everybody is wearing his mask. Everybody is washing hands with soap, and we have also detergents for hand sterilization. This is for myself, for the technicians and the farmers who are working with me in the field. Did you hear me?
I did actually. I think this is the last slide for the Niger presentation, part of the presentation, the near focus, Niger focused part of the presentation. Does, while we have Usman on, are there any questions about the work that he and I tried to describe and what we're doing in 2020?
Hello?
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 Well, I'll go ahead and ask a question quick.
Thank you.
In the past, you've tried to do some assessment of the demand for forage varieties or forage in general. Have you had any interactions with seed companies about your program to develop forage varieties of sorghum? And if so, what has been their response?
Hello? Can you hear me? So
Yes, we can.
I am in contact with, yes, I'm in contact with seed company, particularly Halal and Al-Heri. You know, Halal is for Magaji, who is well aware about the program because he was in INRAN. So they are all waiting. They are putting pressure on us to deliver quickly seed for them, but yet we are not at the end of the breeding process. So I'm still asking them to wait, but they will be involved. I will invite all of them during the farmer field day. So they will actually see the progress we are making. Hello?
Yes.
Good, thank you for that. Other
Hello?
questions for Usman?
Oh, okay.
Well, okay, Mitch, how about we move forward then? I don't see any hands coming up or anything coming in the chat box.
So that's it on our presentation in terms of what have we done in the last project period. And I don't know exactly what we're doing in the last hour. I presume we were doing work planning for the coming year.
Well, great, thank you, Mitch and team. We've taken a couple of pauses for questions on each of the two activities in Senegal and Niger. Let's open it back up for another round of questions from the EAB or from the community in general. Okay, not seeing anything popping up, Mitch.
Looks like Bettina just came off with a mute.
Yes, I wanted to ask, do you have linkages with the Maurice Savagen breeding program?
With Jeff, you mean?
That's correct.
Mm-hmm.
They're all indirect. So these are, we talk with Jeff, but we don't have direct linkages in terms of combined lab meetings or linkages of that type, if that's what you mean.
Yeah, I mean, also when it comes to hybrid breeding, yeah, there should be maybe a regional strategy, I would guess, for heterotic grouping to use the diversity. Is something like this ongoing?
It would be a good idea. We've been leveraging, or we've been using our historical understanding of the heterotic patterns in Niger because we're, it's hard to imagine I mean, I can see it happening in a general sort of way. I can see having OPV forages, but some of the greatest gains in terms of vigor and productivity and biomass yields are going to be in hybrids. And so the main focus of the work that Elizabeth is doing in Senegal is on working in the OPV backgrounds for these new ISRA lines that appear to be widely accepted and liked by farmers in the regions where we've been doing some of the testing. We haven't really talked about pushing into hybrids in Senegal. On the Niger side, there's both the forage or there's both the OPV and hybrid emphasis. A lot of the hybrid emphasis is if we're trying to maximize biomass yields, it seems like hybrids will have to be it's something that at least needs to be explored. And we've been using the historical understanding, especially coming out of Isafu's program, for hybrids that work well in that environment.
Okay. Mm-hmm. And did you figure out what the farmers really want? Like you have all those options, dual purpose, grain sorghum, fodder sorghum. Is all this needed by the farmers?
Usman, can you respond to that based on your survey?
Because I thought some farmers may say we will always want some grains to eat ourselves before we feed our animals. Or how was it in your survey?
We may have lost him again.
He's back.
Usman, can you hear us? Could you hear the question?
I didn't get the last question.
The question is,
Yes.
what kind of products do farmers want? Do they want
Hello?
dual purpose, or where they have both grain and forage, or do they potentially want forage, or do they want to have even grazing options? What did you find when you did your survey?
Okay, you know,
I think what he learned in his survey when he was talking to the what do they call them, the forage traders or the fodder traders, the hay traders, they were very interested in.
hello?
Usman, what did you find in your survey?
Okay, so as you know, before we start this breeding program, I did a survey in Kwani, in Sumarana, and in Tilaberi region. For the small,
We're just having a tough time hearing him, so. Usman, can you,
Challenge.
we can't hear you. Could you type in a response? Or maybe he's gone all the way right now. Looks like he's gone altogether. Usman, can you type a response to the question?
Hello.
I just sent him a chat.
Usman.
yes, I'm tapped. Oh, you
I see.
can hear me, right?
Yes, I can hear you now.
Hello, can you hear me? Can you
Yes.
hear me?
I think he's typing an answer, but I'm not sure.
Yeah, he started to type. I wanted to say that during my survey, he responded to a chat I sent him. I'll let you know if I see something else coming.
I am typing quickly so that you know that I'm typing.
Yep.
Here's another question from Peter.
The small-scale farmers express their opinion. Well, that's for dual-purpose sorghum varieties. So the small-scale farmers express their opinion for dual-purpose sorghum varieties.
Yeah, that's what I was thinking.
For the dairies forage sorghum. Okay, I'm just going to copy and paste it for everybody to see once he's done typing.
Right.
There's another question from Peter in the chat. Is everybody seeing that?
Yes, I see it.
Hello? Hello?
Hi, Usman.
I
Yes.
tried to type quickly, so that's why there was a lot of mistakes. So I want to say that. Hello?
Yes.
Hello?
Yes.
Okay. So I would like to say that for the small-scale farmers, they want the dual-purpose sorghum varieties with specific traits such as high biomass and stay green. But for the dairies, like the one you visited during your visit in October in Niger, their preference is for forage sorghum because they are the one producing milk for industries, like the type of yogurt you ate here. Hello?
Yes, we can hear you.
Okay. So did you get my point?
So your point was small-scale farmers are interested in dual-purpose sorghums because they are interested in both grain for home use.
Yeah, with
And
specific traits for like high biomass and stay green.
and the dairies, the commercial operations, are more interested in what would be in a classical forage sorghum.
Yes.
Peter Matlin asked a question, Usman. He said, if you don't mind me reading it, are there any analysis that examine the economic viability and competitiveness of hybrid forage sorghums compared to OPV forage sorghums in Niger? Especially given the
not
higher,
yet.
especially
Not
given the
yet,
higher
we don't
cost.
have any. Not yet.
And the same question about economic analysis of dual purpose versus single purpose sorghums.
So yet I don't have any economic analysis. What I know is the economical importance for store traders.
And what did that indicate?
It indicates that store trading is actually a profitable business for them. And they were able to demonstrate to me during their survey, because I did a survey for farmers and for store traders. So store traders are having money selling the store they buy for farmers.
Peter has a follow-up question that said, would dairies be interested in dual-purpose sorghums using both feed and forage?
Yes, I'm really interested in dual-purpose using both feed.
Are dairies interested in dual-purpose sorghums, or are they mainly interested in biomass sorghums, Usman?
Dairies are mainly
We're having a pretty tough time on this. One thing that everybody is it's probably worth mentioning. Mitch, if I remember correctly, didn't Usman write a chapter on this in his thesis, and has that come to publication?
Yeah, I forwarded the paper to you. It's been a year ago probably now.
Right, I just can't seem to pull it up. I was looking for it.
When we get off the call, I'll go in my file and see if I can pull it up.
That'd be great. Then I can.
I think that you can go into Google Scholar and pull it up there even.
I just did, and I couldn't find it.
I
just
see.
Canadian forage, so I don't have the specifics of the title. So there is some work. Usman has done some work on this as part of his thesis that we can probably worthwhile reviewing to help clarify some of his findings.
And then Bettina has a question here. I see, what seed yield can a seed producer get with a pure forage sorghum? In many cases, those forage sorghums are going to be harvested at flowering time or shortly after heading time. So the production system is actually doesn't actually even consider grain yield. They're trying to maximize forage quality.
Yeah, but someone needs to produce the seed, isn't it?
Oh, you're talking. This would be mainly for these dedicated forages, which would be mostly hybrids. So in the hybrid seed production, the male and the female parents oftentimes are seed producing types that produce because of the heterotic pattern and adaptation combinations, these massive biomass hybrids that are substantially more productive than either of the parents per se.
Okay.
The OPVs, though, are going to be more in line with what you're describing. And I think actually Usman has data on, I believe they're BC2 or BC3 conversions of MOTA, L-MOTA, and CPON 82. I don't recall the exact numbers. If Usman can remember from his dissertation chapter what the comparison was, they were similar. But I don't think he's on right now. Are there any other questions that I can try to answer?
Maybe I missed it. Are you now also working on sorghum Sudan grass crosses?
Yes.
And can they also be used as a ratoon crop?
Yes. So there would be a dedicated,
in West
it would be
Africa?
theoretically, we don't know the answer to that because we don't see Sudan hybrids there. But we sent him an array of things to look at last summer. One of those is looked like it was interesting and might have some interesting adaptation. It's a very thin stem, narrow leaf grazing type actually. And we had converted that to BMR6. And I promised him that I would try to get, it's an active part of our program. So in our greenhouse production block, I actually have a block of seed increase for a new BMR6 tan plant, sorghum Sudan, that he will be able to look at this summer in Kollo. And hopefully he'll be able to make hybrids and look at those hybrids and see how well they're adapted.
Yes, I think that's something very innovative and interesting to do.
We were actually related to that same question. I'm not sure if Usman is able to communicate this, but we were in, so some farmers in Niger actually use these wild type sorghums as forages. And they're extremely high tillering types. And so while we were visiting the nursery at the Kollo station, there was an example of one of those. There was an example. There were wild sorghums like that in the adjacent field, and Usman is looking at those just in terms of a different project than this, but just from the standpoint of what are farmers doing, and some farmers are using sorghums like that, which would be not dissimilar from these sorghum Sudan crosses.
Mm-hmm. Yes.
So many of these sorghum Sudan hybrids produce 10 to 20 tillers, depending on the fertility and plant spacing, and they're all selected to have very high regrowth characteristics. So they're harvested at flowering, and it's not in a U.S. environment. It wouldn't be uncommon to get several cuttings of what you would call the ratoon crop, and that was really what we were interested in. How would something like that fit in with especially into a commercial animals producer's production system, having an access to a sorghum like that?
I saw this in Kenya on a farm a little bit south of Nairobi, a sorghum Sudan grass cross, and the farmer was cutting it, I think, 3 or 4 times and selling the fodder to other animal breeders or dairies, and was really making big money out of it.
So in my own mind, what I was envisioning is this isn't something you're going to do on the sand dunes in south central Niger away from the river, but this is going to be something when you're down along the river. Smallholder farmers, I can imagine a situation where they might have a couple of hectares where if there's an adjacent or a nearby dairy or another animal producer, or depending on what the forage demand was, they could go in and cut and cut and cut and cut, and potentially have an opportunity to have almost a perennial type of crop, at least for several cuts, where you would have good productivity, especially if they went to the trouble to fertilize it between cuts.
Yes, but you're right. Probably not on the sandy soils of Niger.
Well, like I said, along the river, I can see there's
the
these
river,
highly
yes.
productive areas, but up in the dunes, I don't see that would happen.
Yes, that's true. Thank you.
Any other questions that I can try to help? Or this can either be questions to Elizabeth as well. I think she's still on. Yes, she's on.
I think you alluded to in maybe your explanation, and I'm not sure if this is correct, is Mousman finding support from other donors to look at the forage issue in Niger?
So a lot of what we were doing in our visit was, my point was he's now a sorghum breeder with his own program, and it's dangerous for a research scientist to be dependent on a single source of funding for the work that they're doing. And we talked a lot about what other aspects of the forage program could be developed to attract funding. And he doesn't have funding yet, but he's working on that, and I'm trying to support him in that. And I'm sending Elizabeth and Usman regularly calls for proposals that might fit into their agroecologies and contribute to their professional development.
Great, excellent, excellent support.
In fact, I believe Usman, as soon as this week is over, he has a proposal that that he wants me to help him with.
Great, great, excellent.
Usman, I can see you're there. Do you have anything to say? I'm not sure if you can talk. I know your internet is unstable.
Yes, the internet is not really fine. I prefer writing.
Yep, he says he's developing a proposal now.
Yeah.
So Mitch, this is Peter. On the question of additional funding for this work, is there any potential in the region, not just in Niger, but in the larger region, to get some investment, some funding support from private sector livestock companies, either meat or dairy?
I think that that's going to be a possibility. If nothing else, even if it's in-kind support. I mean, I don't know how much cash support there would be. I don't know the economic status of those animal industries myself in terms of how much cash that they have. But I think that this is going to be something that would be attractive as soon as he has a product that's interesting. If he comes out and says, look, getting back to Bettina's comment a few minutes ago, here's a sorghum sedan hybrid that's extremely high palatability, results in higher milk yields. A farmer can cut it 3, 4, 5 times depending on what they're trying to do. And I'm just making up a story here. And can go talk to commercial partners and say, look, this is our first try at, of these new kinds of products, and I have these other ideas. At a minimum, at least this is what oftentimes happens in the United States. Even if they can't come up with cash, they can come up with in-kind support that makes it easier to write proposals to external donors.
Yeah, no, that's true. I'm thinking primarily of the market in Nigeria, where there's much more demand for both dairy and meat products. I don't know if any similar work is being done in northern Nigeria. Are you aware of any work being done down there that's parallel to what your project is doing?
I'm not. Usman, if you can hear, or if you can type into the chat window, it looks like you're on at the moment. Can you talk a little bit about forage breeding programs in Nigeria or in other countries south of your location in the higher rainfall areas?
I think we just lost him.
Yeah, I guess my larger point, it'd be interesting to get a sort of a sub-regional perspective on how these markets are evolving and where the demand is likely to be, not only in terms of demand for the work that you're doing, but also as a potential source of resource support for this work going forward.
Right. I was actually surprised, Elizabeth, I don't know if she can talk, when we were talking with the farmer groups in Senegal, I knew that forage was important. I didn't know what the ratio of value proposition was in terms of the crop. And if I understood correctly, it was 50% of the money that a farmer might get out of a crop in that environment was from the forage itself. And these are from the types of sorghums, these Isra sorghums that Elizabeth is advocating. One of them is a sweet stem type that the farmers really like because the animals seem to have a strong preference. And he was adamant about the importance of don't just pay attention to the grain, pay attention to the forage as well, because half of the value is tied up in the forage in terms of value proposition.
Yeah, absolutely.
Other questions?
I have a question about what's the value or what's the potential for a sorghum sedan under dryland. So in the United States, a lot of sorghum sedans are grown in dryland environments, but they're different than the dryland environments that we see in much of West Africa, where the duney, I don't know, the more dune-like environments across southern Niger, a lot of the productivity is constrained by fertility. And in a lot of the dryland environments through the south central part of the United States, the soils can be quite fertile, but they can be constrained by water. And so oftentimes in those environments where fertility is not constraining, it's possible to actually have these sorghum sedans are well adapted. I think I saw Tim Lust on here earlier. I don't know if Tim wants to talk a little bit about adaptation of sorghum sedans across the area where he's at in the highlands of Texas. But I'll speak. Oh, here he comes. Yeah.
Yeah, I can just say that there are some that are focused certainly more on drought tolerance, while others, as Mitch said, are really more focused, from a nutrient standpoint. But there is a wide variety of genetic material that falls under that sedan grass category. There's one for everybody.
Thanks, Tim. Any other questions? I'm looking at the chat. You can chat to me, or if you have a decent connection, I can try to answer the questions.
So while we're waiting, I wish Usman was able to answer this, but it was about 1.5 years ago. I remember talking with the sorghum breeders from ICRSAT working in northern Nigeria, and they did not have a program that was looking at forage. So back to your question, Peter.
They're not having a program on that. I'm not sure what that tells me, but.
That's just one source of a regional.
Sure. Yeah. Well, it was. Yeah. But they're very stretched for resources, so they have to be very focused on a certain set of priorities that they're most comfortable with delivering against.
Right. Usman, while we have you on, if there's no more questions, give you any last things you want to contribute before you phase out? Elizabeth?
Elizabeth has said she has a weak connection. If you can chat it as well, if you can't say it, Elizabeth.
Yeah. I don't know if you can hear me.
We can hear you.
I've been able to follow all presentations from day one. I don't know why tonight my Wi-Fi just decided not to work. So I'm using my data. Hopefully that won't stop anytime soon.
We can hear you well.
Oh, okay. That's good. So I think, wait, did I miss? There was a question for me?
The question was, you have an opportunity to say anything to the group about what you've done so far and what you see moving forward.
If I see an opportunity?
in terms of the work
It's
that's going to
breaking.
happen in 2020, 2021?
I think it's just breaking. I don't know if you can type the question. I hear parts. So,
I'll send it to
this
you.
seems to be a Senegal thing. I thought I was the only one. Okay.
Well, I think what we can do at this point, if you can hear us, both for Elizabeth and for Usman, is that we can take any questions and we can forward them on for an email response. Seems to, as Njito is indicating, the network seems to be terrible right now in Senegal. Everybody's home probably accessing it, whatever the case may be. Mitch, do you want anything last to say before we conclude this session? Cliff, Mitch?
For me, one of the things that I've been trying to spend time, this is this particular project, the main focus, I mean, we've been talking about the project in Senegal, the project in Niger, but if you step back a little bit and look at what are we doing here, we have two new PhD students, or they're not PhD students, two new PhD scientists that are initiating programs in West Africa. And one of the major and extremely important goals of this project is to really support their professional development and support the development of these programs, because in both cases these are new programs. These aren't necessarily stepping into that this is a program that's been around for 20 or 30 years, and of course we need one of those because we've always had one. I was impressed, for example, when we went to talk to the DG in Niger, how excited he was to hear about the work that, this was the last day I think I was in Niger, either the last day or second to the last day, how excited he was to hear the work that Usman was doing, and how he wanted to hear more about the work on a regular basis. And he thought it was really exciting because this is something that he always hears from his government colleagues, that what new is happening at INRAN, and he was excited to be able to go back to them and say, well, we're developing this new forage breeding program in the case of Niger, and we've got this fresh new phase PhD, brand new freshly minted PhD, who's got a supported program and some great ideas and seeing some success. The same can be said for Elizabeth. While I was there, they actually had a regional, a countrywide, country level meeting on planning inside. I'm not sure, it was probably an ISRA type meeting. And the same comment of, we've got these new students, whether it's Cyril or Elizabeth in Senegal, how do we support their programs moving forward? How do we provide professional development? Because it's easy to say, here are some funds to get something started on a SMIL2 project, but as you all know, professional development is much more than a project by project activity, and without a lot of mentoring, without a lot of support, these programs will just fade away for lack of consideration. And I think it's really important, and it's something that our project is really trying to take to heart. How do we develop strong scientists and leaders in the region? Yes, they have some support from SMIL2, but in a larger context, how do we support them as they develop a program of their own and a reputation of their own? And a reputation, so you can look at it from a SMIL perspective, that's going to be rooted in all of the great opportunities that they had in SMIL, so that they can look back and say, I started off as a PhD student supported by SMIL. I got started in my home institution supported by SMIL. SMIL helped me develop these new ideas and this new germplasm and new preliminary data with help from, sometimes it might involve someone like me stepping in, or it might be someone else on the team, or it might be, I think one of the things we have to keep thinking about moving forward is how do we support their professional development? That's the main thing I think I wanted to talk about.
Great. I think that's a good question to continually ask.
Cliff?
I would have seconded that, actually. That was the thing I was going to say if you didn't. So I think it's also really important to see this as a way to kind of expand the uses of some of these things too. So Elizabeth was mentioning the use of these as poultry feed, and there are other aspects of the grain that can help in that too, with regards to starch digestibility that can be combined with the protein digestibility. And those are things that we can start to explore as well, since she sort of identified a slightly different market than the one we might have originally thought. So I think it's really good that they're starting these labs, but also that they're expanding the range of the uses for these things even beyond what we had thought of. Did any of that come through, or did I just mute
We got
myself?
you. We're just waiting.
Great job, Cliff.
Okay, I was afraid I had actually hit mute and not unmute.
And Gino contributes that there are industries looking for viable options for poultry feed, large-scale poultry industry.
Khalil, there he is
Yes.
again. Look at that.
Yes, can I comment?
Yes, you can, Elizabeth. We can hear you.
Yeah, so one of the industries that we met, like Sedima, is actually interested in that. They do formulate poultry feed, and they actually was telling us they use up to 7 tons per day in terms of cereals, all cereals combined, and actually they rely on importation to get their cereals in. So when we show them some of the figures that we have based on the sorghum varieties that we are working with, they were excited because they did not know actually that they could get some of the nutrients that they're looking for just on sorghum, on local varieties here on sorghum. But still the demand is high. As they said, they use 7 tons per hectare. We need a large scale, and we are not actually, we just developed those varieties. So hopefully they can work with, how can I call it, seed production industries to meet their demand. But what we can provide is the varieties that are high yielding and also have good nutrition quality. So that's why, as I said earlier, it was important to initiate those discussions, let's say 2 years before even going to present a product that is ready, for them to actually know that there is something that is being done in the country that they can benefit from and not just rely on importation, which is really a high cost that they also mentioned. So, yeah. And also I wanted to add to what the partners at Purdue were saying. I think this project is very important in the sense that it meets our it helps respond to some of the, how can I say, questions that we have in our breeding programs. It's not like SMIL is actually the main activity of the SMIL activity are the main activity that we have in the programs, in sorghum breeding program in general. We have a breeding product profile that we try to meet. So based on the project that we have, we see how they can help in meeting some of our objectives that we already have. So that's how we're working with the projects that we have. So as part of the results that we have with the activities from SMIL, they help now build another, how can I say, not another program, but as a base to get more funding, for instance, to look at some of these aspects that we're looking at, like integrating forage breeding program in our program. It's not like we were not looking at it before, but in addition to feeding, to improving these lines for protein digestibility and other essential amino acid content, if we have also if we meet the farmers' demand for the forage will be also ideal. So these are things that we look into in other funding, actually.
I see Usman is on. Usman, I'm going to try to give you one more chance if you can say anything about your research program to the group.
So what I can say is that up to now we are having good progress. People, particularly dairies and big farmers, are still coming at INRAN looking for varieties with high forage potential. But unfortunately, at the INRAN seed unit, we are still giving them SIPON 82. And I know that SIPON 82, yes, it is a good line, but still it doesn't have the requirement for forage quality. We are giving them SIPON 82 because the lines we are breeding for are not yet ready. And INRAN, before my PhD program, doesn't have any brown midrib or hybrid sorghum Sudan grass line. Now we are almost at the end of the process. Very, very soon, I think we will, with the promising line we have, we will meet the demand people are coming to ask.
That's excellent. Thank you.
Yeah.
Very good. Thanks, Usman.
Because, yeah, thank you.
Tim?
Okay, it sounds like we've come to an end of this session. Thank you, everyone, for participating. It's been a rich discussion, a little bit struggling with some of the connectivity, but that's not to be unexpected. I'm going to close this session.